of list stuff coming up again in this lecture. Okay. So, all right. Thank you very much. Okay. I never told you that that we're in my area now. I was an amino acid chemist, okay, and I worked on amino acids and amino acid synthesis in CNS in mice and guinea pigs, okay. This is all the learning objectives. Okay. All right. Now, this looks complicated, okay, but we've already started to deal with it in some way. This is the TCA cycle, okay. This is actually ketone body formation, and we're actually looking at how amino acids are degraded, okay. All right. Now, important, we're going to want to know whether they're ketogenic or glucogenic. This, you have to be able to look at it because, for instance, if I have an amino acid that goes to alanine, okay, under conditions where you're not gluconeogenic, alanine can go to pyruvate, and pyruvate can go to acetyl-CoA, correct? All right? Everybody see that? So we have to always determine it under conditions of gluconeogenesis. So what would create gluconeogenesis? What hormonal release? The absence of or the lower levels of one hormone and increased levels of another hormone. Come on, don't be shy. What? Insulin and glucagon. So you want low insulin and high glucagon, so you can do it with insulin. So it has to be, so under those conditions, because then under those conditions, pyruvate can only go to oxaloacetate. It can't go to acetyl-CoA. Okay. Let me ask a question. Are fatty acids, even fatty acids, gluconeogenic or ketogenic? I'm asking you, my question was asking, I was very specific to say even chain. What's the end product of a breakdown of fatty acids? Acetyl-CoA, all right? So under normal conditions, not under terms of gluconeogenesis, the acetyl-CoA, can it go into the Krebs cycle? Yeah, absolutely, okay? Yeah, right, that's true, okay? But what happens is, why is it not gluconeogenic? Well, because they don't show you here, but there is a isocitrate is here, and it loses a CO2 here, and alpha-ketoglutarate loses a CO2 here. So what happens is, acetyl-CoA goes in before the CO2s go out, okay? And so they're not, so the carbon that you put in is going out, not necessarily the exact carbon, but effectively the equivalent of that. So you can't make any net glucose. You can't make any net OAA from acetyl-CoA. So fatty acids that are even chained produce acetyl-CoA, and so they can't be gluconeogenic, okay, because they enter the Krebs cycle before the CO2s come out. Okay, so if you go back to your Krebs cycles, which is a little more detailed, you'll see the CO2 comes out here and the CO2 comes out here. Okay, but they're decarboxylations, okay? Is that making sense to people? Yes, sir?
Wait, the CO2 that comes out
isn't exactly the same carbon that went in. But the problem is that with the second cycle around, most of the CO2 that went in, so you're not making any net. So if you use labeled acetyl-CoA, you would actually see the label, but you wouldn't see any net change in the amount of glucose that you produced. Okay, so it can get incorporated, okay, but you can't, it gets incorporated, but you don't make any net, because putting in two carbons and getting out two carbons, in essence, no. Yes, ma'am? Absolutely, yes, that's the point I wanted to make. The odd chain fatty acids, okay, would be, and what pathway do you think they were going to be, where do you think they would go? Right, exactly. Propionyl-CoA from odd chains go that way. And we have this group of amino acids, the valine 3, with the ones I call the vitamin M okay, they can go to propionyl-CoA and go to succinyl-CoA, so they are gluconeogenic. So the problem is most of us don't eat a lot of whale blubber, which is aquatic mammals have a lot of odd-chain fats, but they're not. We probably have this pathway, at least we're now, primarily to handle these amino acids, okay, but they'll be gluconeogenic. So you've made the point. Thank you very much for doing that, okay? Everybody, is that clear to everybody? Even chains, no, under any conditions of gluconeogenesis. Odd chains, yes. And even odd chains, if you were metabolizing not under gluconeogenic conditions, okay? So they could be used for energy, okay? So, all right, important things. So we're at gluconeogenesis, okay? So anything that goes to pyruvate has to go to OAA, and then that goes off to glucose, okay? Everybody comfortable with that? All right. What about ketone bodies? We under gluconeogenesis, do we make ketone bodies? I can't make glucose without making ketone bodies. Why? Because I'm, what?
I didn't know that.
Now you do.
Why? Because
But think about, okay, we're going to make a glucose molecule. What are we going to make it out of? No, what are we using fatty acids for? The energy, yes, okay, so right, now you know why I looked at you that way? Okay, where's the carbon structure coming from? What are precursors of these amino acids? Okay, those amino acids, the carbons come from the amino acids, okay? They're not going to come from carbohydrates, right? Because we're low on carbohydrates, so effectively the only thing that they have to come from amino acids and a few other things, some pyruvate or something like that, okay? If you got it from lactic acid or something like that, right? Okay? So that's really the issue. And so the energy is coming from the fatty acids or going to acetyl-CoA. Yes, sir?
I don't understand why when acetyl-CoA comes from an amino acid, it's considered gluconeogenic.
No, it isn't.
Oh, it is?
So what's happening is, under gluconeogenic conditions, the pyruvate it can't go to acetyl-CoA. Actually, the enzyme is inhibited, okay? And effectively, it can only go to OAA. It doesn't have a choice. That's why I'm telling you the only way to see this is under gluconeogenic conditions, high glucagon, low insulin, okay? Is that making sense? It's a good question, okay? All right?
So the acetyl-CoA only comes from fatty acids in that case?
They're only coming from, yeah, and what's happening is all the OAA that we have is going to where? To glucose. So what's the first enzymatic step in the Krebs cycle? Citrate synthase, right. As remember taking an acetyl-CoA and OAA. Well, guess what? I don't have any OAA in the liver. It's going off to make glucose. So what's going to happen? In the liver, I said. It could be in muscle, okay? What's going to happen? What's going to accumulate? Acetyl-CoA, right? Yeah. And so there's a limited amount of acetyl-CoA and CoA in a cell. So what's going to happen in that cell if I start going into gluconeogenesis? I'm going to all of my coenzyme A is going to end up as acetyl-CoA, right? So what happens to the fatty acid oxidation? It stops. And that's where I'm getting what? FAD and NADH from that part to, in essence, power the gluconeogenesis to make the ATP that I'm using in gluconeogenesis, right? Is that making sense to you? Okay, so are we clearer now on that? These are good questions because you're asking questions that kind of tie things together, okay, and make sense, all right? Everybody kind of comfortable with that? Excellent response, by the way. That was really an excellent question, okay, that you were asking it. Okay, so what I said to you is, when we were talking about the amino acids, I said, remember the number of carbons in there, okay, and so that we can do it. So for instance, we'll start with these. These are C3 amino acids, okay? The C3 amino acids go to pyruvate, okay? That's what you need to know, okay? All right, you don't need to know anything else about them, okay? And if they go to pyruvate under gluconeogenic conditions, are they gluconeogenic or ketogenic? Gluconeogenic, absolutely. Okay, so those are three, yeah.
I just realized, like with ketogenesis, like if we break it down to create acetyl-CoA, but then acetyl-CoA activates pyruvate carboxylase.
Mm-hmm.
So is that the connection there? So it's just like I just wanted to share that.
Yeah, yeah, that's what it is. The enzyme is it's activated by glucagon. It's activated by acetyl-CoA, and you're shifting it in that direction. Okay, it's a high acetyl-CoA, and so what happens is that the ketone bodies form. Why are you making ketone bodies? You're just not. You're trying. The liver needs to have the fatty acid oxidation, and so what's happening is the ketone bodies are giving us a huge amount of of coenzyme A. I don't have enough to take the fatty acids and metabolize them, and therefore I don't have any energy for gluconeogenesis. The process will stop. Okay, but that's a good point too, though. Okay, that the acetyl-CoA activates pyruvate carboxylase. Okay, all right, good. All righty. So we can go with three. Actually, glycine actually goes into this group. If you kind of remember, you can make when we did the folate and we were doing serine hydroxymethyltransferase, we got glycine out, and glycine could go in. It could go back in the other direction. So it's pretty much the same way. Here, okay. The next number up is effectively aspartate and asparagine, and they're C4. They're going to go to oxaloacetate, which is also C4. Okay. That's coming down here. We're going to come here, and glutamate is 5. Okay. And glutamine and proline are 5, and these two, arginine and histidine, are 6, each 6 carbons. So the 5s and 6s go to glutamate. Okay? Making sense? Okay, so that's easy. All right? Now we have the vitamin M group, which gives us succinyl-CoA, which is valine, isoleucine, methionine, and threonine. What happens is that we start with isoleucine. It's a branch chain, which we'll see, and you have to go through another enzyme to get to that stage. So there are other things in here, but effectively, if everything's working right, it'll go to propionyl-CoA, which we'll see in a little bit. Okay? All right? So these are good. This is an aspartate that we're going to produce, or when some of the enzymes. These are ones that are pretty much the same way. Tyrosine and phenylalanine and tryptophan are aromatic amino acids, okay? And so they are going to be mixed, and part of their molecule is going to go to fumarate, and part of their molecule is actually going to go to other precursors, and we'll see that later on, okay? And then these are the ketone bodies, and we'll talk about that in the next slide. Things that go to ketone bodies, HMG-CoA, and you make ketone bodies and do it the same way. You then send the ketone bodies out of the liver, and they go to the other tissues, and then they're reconverted back to acetoacetyl-CoA, and then that's broken down to acetyl-CoA. And since it's another tissue and it's not the liver, there's still plenty of OAA for it to be used for energy in the other tissues, right? Because the OAA isn't going out. Remember, gluconeogenesis is only in the liver. Okay? All right, let's take a look. Okay. So we start with the easiest list. There are only 2 that are purely ketogenic. Leucine and lysine, the 2 L's, they're both 6 carbons. No reason to remember that because you don't have to worry about it, okay, at that point. These 2 amino acids go to ketone bodies, okay? Everybody see that? So they're ketogenic, okay? Okay, the glucogenic only, 13 of the amino acids. So right now we've accomplished 15 of them, okay? All right, so far, okay? And we'll go through these, okay? So glycine, alanine, serine are 2 and 3 carbons, okay? And these 4 are converted to pyruvic acid, which goes to glucose. Everybody see that? Okay, it's in your syllabus. All right, aspartic acid is 4, asparagine is 4. These 2 amino acids go to oxaloacetate, which goes to glucose. All right, pretty much the way as it is on the diagram. Okay. Glutamic acid is 5, glutamine is at 5, proline is 5, arginine is 6, histidine is 6. The 5s and 6s all go to alpha-ketoglutarate, which goes to glucose. This is not in your textbook. This is something I put together so that you could actually have it in writing so that you can remember it that way. okay? Methionine is 5 carbons and valine is 5 carbons. These 2 amino acids are converted to succinyl-CoA, which goes to glucose, okay? There are 2 others as well, okay, and we'll deal with it that way, okay? Okay, now mixed glucogenic and ketogenic are 5 amino acids, okay? So we had 2 that were ketogenic, 13 that were gluconeogenic, okay? And now these are mixtures of both, and they can do it a couple of different ways, okay? Everybody kind of see that? Okay. Type 1, during catabolism of these amino acids, and they are the aromatics, which is phenylalanine, tyrosine, and threonine, part of the molecule goes to glucose and part of the molecule goes to ketone bodies. So they're producing both on that basis, okay? So every time you break down a phenylalanine, you're going to get something that goes to the ketone bodies and something in the other half that goes to glucose, okay? Okay, so they're both. Not a good, if you wanted to get rid of somebody was having problems with ketone body formation and a gluconeogenic, okay, could be a problem, okay? So there's the number of carbons, not terribly critical. They have a lot of carbons, okay? Don't worry about the number because the aromaticity will tell you what's happening in that. So that's type 1. This is type 2. Amino acid isoleucine has 2 different catabolic pathways. One goes to glucose and the other one goes to ketone bodies, okay? So that's what happens with isoleucine, which is 6 carbons, okay? 2 different separate pathways. So it's both, sometimes some molecules go to glucose and some molecules of isoleucine go to ketone bodies. Type 3 is this amino acid, threonine, has 3 different catabolic pathways. 2 go to glucose and 1 goes to ketone bodies, and it's 4 carbons, okay? Is that clear? I think it should help you know. For the most part now, we've covered catabolism of amino acids. You know what you start with, and you know what it goes to, and whether it's ketogenic or glucogenic, okay? So the intermediate, it's good to know that it goes to, in this case it doesn't, you don't need to see it. You'll see it, it's the T, threonine is the T of vitamin M, okay? So it goes, and if you wanted to add in, it goes to succinyl-CoA. Okay, by 1 pathway. And this one is direct. Okay? Any questions? Okay. So now what we're going to do is we're going to look at, and now these are ones that I selected, amino acids breakdown. This first one is methionine. It's actually the breakdown of methionine, okay, on that basis, okay? Synthesis and degradation of cysteine, okay? And the reason we're looking at this one is that we have a couple of inborn errors of metabolism, okay? And we want to see what's happening, okay? So when methionine loses its methyl group, it goes to, remember what's the cofactor form of methionine which is used for methylations called? What are 99.9% of the methylations are carried out by what cofactor? Methylation. The methylations are only one folate reaction is carried out. Methylation is carried out by folate. That's hydroxymethyl. Methyl, yeah. What? SAM, acetylmethionine. That's the methyl group. And then what does it go to? SA, right? Right, it goes to SA. It's acetylhomocysteine.
So what's the point of the
N5-methyl-FH4?
Yeah.
To work with methionine synthase, which is really regenerase, okay?
So it's not, what group is it?
It only regenerates methionine.
Oh, that's really what it does.
It's the only thing it does. The only thing, it's only one reaction. It's the only thing it does. Are there questions over there? Okay. Yeah. Excuse me.
With the methylbutyl and methylfolate, how does it, I thought that's just where the methyl group is coming from the homocysteine, but is that just for...
No. So it's what's happening is if we looked at the structure of methionine, if I put a methyl group, the methionine would have a methyl group here, okay? When it in SAM, it's associated with adenosine, okay, and that's the SAM, okay, all right. S-adenosylmethionine is what it is, okay. When the methyl group is given up, correct, what we have is SA, just homocysteine on the left, okay, which is 4-carbon cysteine analog, or homolog, okay. Cysteine is 3 carbons, and homocysteine is 1 longer, okay. It has another methylene. Is that making sense to people? Okay. Yes? No? Yeah, good. SAM does 99.9% of methylation reactions. It methylates nucleotides, it methylates RNA, it methylates all kinds of neurotransmitters, okay, and all of these other things, okay? All right. So let's call it, we want to call this the methionine breakdown, okay, which is catabolism, okay? So methionine loses its methyl group, it's given it off to somebody else, and I end up with homocysteine, okay? You can see, all right? Now, what happens is that this combines with a serine molecule, which is C3, in the presence of a cystathionine synthase, okay? And it subtracts water, and it takes an OH from here and an H from there, okay? And it gives us this large molecule, which is serine and homocysteine. That molecule is called cystathionine, okay? All right? This is then broken down by cystathionase. This is a PLP enzyme, this is a PLP enzyme, okay? So pyridoxal phosphate, okay? It's an amino acid. And then what happens is that this is then broken again, and the sulfur ends up on the 3-carbon, okay, which is then cysteine, and the carbons then come out as alpha-ketoglutarate, and they go into the propionyl-CoA, which is the odd-chain fatty acid pathway, okay, and dealing with it that way. So in this breakdown of methionine from methionine isn't recycled, right? So the only time is if I end up breaking down homocysteine, I have to use N5-methyl to transfer the group back to cobalamin and effectively do remethylate it. So it's not a synthesis of methionine, okay? What it really is the homocysteine is the essential thing because it's the one that contains sulfur, okay? And effectively, we can synthesize cysteine, which is a non-essential amino acid. We make it this way, and don't worry about this, but we've made cysteine, okay? All right. The real problem that I have is can have a deficiency disease of cystathionine synthase, okay? Which brings about what? What's going to accumulate? If I have a deficiency of this enzyme, what will accumulate? Homocysteine, and you get then what's called a homocystinuria or a homocystinemia, meaning it's in the blood, okay? And it's been shown that homocysteine in the blood has some detrimental effects on the CNN, on the heart and heart system, okay? So high homocysteine is a problem, okay? So you don't want to have that deficiency disease, okay? All right. Okay, and then we don't make cystathionine, so I don't make cysteine. So what happens to cysteine? Is it now an essential amino acid? Yeah, absolutely, because I don't have a way to get the sulfur onto it, okay? Because I have it made, I can only make it by that one pathway, okay? All right, there is also a deficiency disease. So this one is called, if you wanted to present it, would be called homocystinuria, is the way the disease is described. We'll see that later, okay? And now there's also a deficiency of this enzyme, cystathionase, which makes cysteine and alpha-ketoglutarate. The real problem then is that we end up excreting cystathionine as well. So that disease is called cystathionuria, okay? And that's the other deficiency disease, okay? If I had same issue, if I have only this one missing and not this one missing, I still end up with a homocystinuria because this is accumulating, okay? And cysteine still becomes an essential amino acid, okay? You have to make sure that cysteine is there, okay? Everybody comfortable with that? Okay, not too fancy, okay? I'm dealing with it. Okay. Okay, here's conversion of amino acids to succinyl-CoA. We just kind of looked at what happened to, here's methionine going to SAM. That's donated. We get S-adenyl homocysteine. That's then cleaved to homocysteine. Serine comes in, and this enzyme is cystathionine synthase. And effectively, we make cysteine, and this is a PLP enzyme, so is that, which we said before. And alpha-ketobutyrate, and the alpha-ketobutyrate can go to propionyl-CoA or whatever. Threonine can come in here and make alpha-ketoglutarate by threonine deaminase. Remember, it's one of the few that deaminates directly, okay? And isoleucine can come into propionyl-CoA, and valine can come in the same way. So these all go to succinyl-CoA, okay? There's some repetition in this, but we've added on this, and that. So this is that way. Everybody kind of comfortable with that? Okay. Okay. Here we now have probably the one enzyme, the one amino acid that kind of works overtime in terms of inborn errors of metabolism. Tyrosine is an essential amino acid or a non-essential amino acid? Don't all answer at once. Loud enough so I can hear it. It's a non-essential amino acid. It's actually made from phenylalanine. Okay? All right? So here we have phenylalanine. Okay? And phenylalanine goes to tyrosine. And we're going to see that this is a hydroxylation of this. Okay? And it's an interesting dehydrogenase, an interesting oxidase, okay? All right, so here's phenylalanine, okay? It's going to combine with a molecule of O2, which is oxygen, and in the presence of phenylalanine hydroxylase, it's going to give us one of the atoms of the oxygen is going to go into water, and one of the atoms of the oxygen is going to go into phenylalanine and give me the hydroxyl group, which gives me tyrosine. So this is the synthetic pathway for tyrosine, okay? It's part of the breakdown pathway for phenylalanine, which we'll see in a minute, okay? Once again, I am not using in this case, I have a reductase. The reducing agent is tetrahydrobiopterin. It donates 2 of its hydrogens, H2, and it gets converted to quinonoid, which is called bihydrobiopterin. Biopterin, thank you. Thank you, ma'am. Okay, and then what happens in this situation is, once again, just like we did with thyroidoxin, NADPH is reduced by, in this case, dihydropteridine reductase goes to this and regenerates BH4. We're going to see BH4 showing up again in serotonin synthesis and others. So it's just another one. Effectively, it's a reduction, okay, on that. Everybody kind of... So I wouldn't worry the steps. Just know that phenylalanine has to be hydroxylated with this. Oxygenases or oxidases that function this way are called mixed function oxidases. The way you can tell them is they use molecular oxygen, that's that, okay, as opposed to O, all right? And they also, they use molecular oxygen, and one of the oxygen goes into water and one of them goes into the product, okay? Not both of them go into one molecule, okay? It's called a mixed function oxidase or an MFO. Okay. All right, so let's go back now. Okay. So this step is phenylalanine to pyruvate. The enzyme is phenylalanine hydroxylase. This is the first inborn error. PKU, if you read your soda cans, if you use anything that has sweetener in it, and it has which sweetener do you usually see in soda cans? In artificially sweetened. Does anybody know? You don't read your soda cans? You should find out that there's benzoic acid in them. Yeah. What else?
Like, there's a sugar with phenylalanine in it. It's called aspartame.
It's aspartame. It's a small peptide that has phenylalanine in it, okay? And so you got a real problem with somebody who can't metabolize phenylalanine, okay? That disease, does anybody know what it's called? This deficiency of this enzyme? What's the PKU stand for? Phenylketonuria. So it says on the soda cans if you're having aspartame as the sweetening agent, okay, which is very common. By the way, it doesn't have a long half-life when you're storing it. But if you've ever had a bottle of soda or a can of soda that has aspartame as the sweetener, after about 3 months, it doesn't taste sweet anymore, and that's because the peptide breaks down. So it doesn't have a strong one on that basis, okay? So phenylketonurics can't eat that, okay? Now, the deficiency of the PKU has been known for a long time. So what do you think you would do in a patient that had this problem of not being able to metabolize phenylalanine? How do you think you'd treat it? Choices are removal of phenylalanine from the diet. Okay? You're saying yes? You're wrong. Why? Okay, you got to think of another area in another box. You want to limit the phenylalanine in the diet. And the reason for it is that, what do you know about tRNAs loading? Suppose I have a tRNA that doesn't get filled. What happens to protein synthesis? What happens to protein synthesis? Do you remember protein synthesis? Yeah, I know you'd had it. What? It stops. Why? It has to have all the tRNAs have to be loaded with an amino acid, right? Or phosphorylation, essentially making peptides or proteins aren't going to happen, okay? So you can't do it. So you have to put enough, or leave enough phenylalanine in the diet so that you will maintain protein synthesis. And it's the same for when we're going to see the maple syrup urine disease, there's actually 3 amino acids. You can't completely limit them because the protein synthesis will stop and the person will die, okay? So you have to have protein synthesis. Is that clear? All right. Thank you for answering the question, but you'll learn better. You see, but you always have to do it. So you limit it. You don't remove it, okay? Because you've got to have protein synthesis going on. Okay. All right. So that's the first one, phenylketonuria. All right. Then what happens to tyrosine? By the way, phenylalanine can go over here. It can actually go into making melanin. We're going to see that I think this is the last lecture on next Friday. Okay. And then, in essence, what's happening, and we have this, and it puts another hydroxyl group, aminotransferase, I'm sorry. The amino group gets transferred to a carbonyl, and we end up getting para-hydroxyphenylbutyrate, okay? What happens is there's a deficiency of this enzyme, and this one gives you a deficiency of this one, the aminotransferase. It's this one. Okay, it gives you a tyrosinemia 2. So what it's saying is you're going to get tyrosine in the blood, and it leads to neurological defects and may lead to lesions of eye and other organs. Okay, on that basis. So that's the deficiency disease for this one. Okay, now we get para-hydroxyphenylbutyrate, and that gives you don't have to worry about it. You get this intermediate, which is homogentisate, okay? A deficiency of the oxidase for this gives you a deficiency disease called alkaptonuria, okay? Alkaptonuria, and I'm going to tell you the way I told it to a class a couple of years ago, and then I had to explain something, okay? All right? So what happens is that your urine turns black on standing. That's the way I did it, okay? So your urine turns black on standing. And somebody raised their hand and said, what happens if you sit down on the toilet? And I said, the problem, it's not a postural issue. It's actually a deficiency disease, okay? And that if the urine is if you don't flush, you know, it shows up as dark, or it was picked up on infants' diapers, and that there was black staining. It's a relatively benign disease, certainly in children, okay? There doesn't seem to be any real problem. In people who have had this deficiency disease, and this is one of the ones that the state of New York and many other states actually test for, okay? They find that it actually ends up in joints, black in joints, and may cause some kind of rheumatic or arthritic issues on that basis. So this is a natural, but it's not a postural deficiency. Okay, all right. So you have to be very careful whenever you say these things. Okay. Okay. Then there's another tyrosinemia here, which is a deficiency of this enzyme, okay, which gives a tyrosinemia 1. I have a feeling it's 1, even though this one is 2, is that this was seen first, so you're not handling tyrosine for some reason. And on this one, and this is in the marginalia in the amino acid thing. If you look up tyrosinemia, you'll find in your index. I hope they have an index in the 5th edition. Can you get to an index? What? Oh, the 4th one, yeah.
The index is fine for any edition. I don't know if there's an index in the online version.
Yeah. But if you look at the chapter in 5 that has the tyrosine, it'll ask tyrosine breakdown or something like that, or catabolism. Okay, good. All right. So this one is it's a transient tyrosinemia. I'm sorry. It's a tyrosinemia 1. It gives you liver failure and early death in the first year, and interestingly has a characteristic smell. Okay, I assume from the urine, a cabbage-like body odor. Okay, I assume that's like probably cooked cabbage or cooking cabbage. Okay, on that, which has a strong smell. Okay, on that basis. The other one is PKU. They will describe the smell of the urine on the. These were picked up by neonatal nurses. Okay, and we'll see the same with maple syrup urine disease. The smell is described officially as mousy. Okay, now it's not like I described that rodent smell in the 168th Street subway. Okay, which was the hypurates and stuff. Okay, that's a rodent sort of smell. Mousy smell is the smell. I actually, for some reason, they were testing questions on national board questions, and this is probably 25 years ago, and they were doing scratch and sniff on the question. Okay. And you had to, this was for the national boards. At that time it was called the national board exams. You had to scratch and see what describe what it smelled like, okay? Was it mousy, or did it smell like maple syrup, or any of these things? It was a scratch and sniff. It never got into the national board exams, okay, on that basis, so, okay? So those are some of the ones that we see with the deficiency diseases, okay? So PKU is a problem. Certainly homogenetic aciduria is not as much of a problem. And the tyrosinemia is a real one, is a big problem on that basis, okay? All right. I think we're almost downhill. And we should finish in a little time. Well, we have one more that we really have to see. Okay, this is the degradation of the branch chain, okay? Here's the stuff we gave you before, propionyl-CoA, and goes to gluconeogenic, okay? So valine, isoleucine, and threonine, they're all branch chains, meaning that they don't have straight chains, okay? And what happens is they're converted into keto acids, okay? And what happens is they are then worked on by an oxidative decarboxylation alpha-ketoglutarate dehydrogenase, okay? Which produces CO2 and NADH, and for all of them, okay? This enzyme, whether it's working here and here, actually belongs to the vitamin pill group. The vitamin you have, if you look on the list of lists, it says the vitamin pill enzymes, and they have lipoic acid and niacin and all of those in there. Okay, the two that you've had so far before this were pyruvate dehydrogenase, right, and alpha-ketoglutarate dehydrogenase. This is the third and only other one. Okay. For instance, what were the 5 vitamins that were used in that? Anybody remember in the vitamin pills? Thiamine pyrophosphate, thiamine, right? NAD, niacin. What? Pentathoic acid, very good. What else? What? Riboflavin, okay. And I think we got them all. 4 or 5, okay. All right, so those are the ones. What? Thiamine is really important. So for instance, alcoholic mothers with children, okay, with giving birth, will give a false positive for their diet is low in thiamine because they're getting most of their calories from distilled liquor, so they're not getting enough thiamine. They will give a false positive for maple syrup urine disease, okay, which is what this disease is. A deficiency of this is called maple syrup urine. It's picked up by neonatal nurses. I don't know when, where, and effectively it inhibits this enzyme, and then these go off to making chemicals and things enzymatically and non-enzymatically, which smell like maple syrup or burnt sugar, okay, on that basis, okay. If you've ever been making flan from, I'm not a flan baker or a maker, but you make the caramel of flan by heating sugar, and if it gets you get a phone call in the middle, you end up burning it. That's the smell you want, okay? So this is maple syrup urine, okay? So these three branched chain amino acids make up most of the amino acid population in many tissues like muscle and all of that. So now, how would you treat them? Now, let somebody on this side of the room tell me, how would you treat them in this case? I mean, the real problem with maple syrup urine disease is you get mental retardation from the products, not just that your urine smells like maple syrup, okay, or you think you're in an IHOP, okay, on that basis, which they don't use real maple syrup. We're talking about real maple syrup as opposed to artificial maple syrup, okay. Yeah, what do you think?
My response is going to sound really crazy. Is there a way to get the branch chain directly into the muscle, without just intravenously?
No. So anybody, what would you do? You would limit these in your diet. You would have to give them a homogenate of protein, okay, which had low amounts of these amino acids, okay, and that's the way to treat them, okay. Not a great treatment. You're not curing it. You're just treating it and hopefully lowering it on that basis. Okay. Any questions? Okay, where do they go? All of them end up going to, they're gluconeogenic. They go to succinyl-CoA, okay, under gluconeogenic conditions. Everybody see that? Any questions about that? I don't know how many they test for now. I don't know if anybody knows. How many tested diseases do they test for? I think it's over 50. Yeah. At birth, they test the urine, and is it more than that? Maybe 100. Yeah, but these are the ones that we talked about are clearly tested for. So okay. This one we don't have to reboot. This is histidine, and it's the one. Histidine is broken down by histidase, and this is the Fig-Lu pathway. And what happens is these carbons, 5 of these carbons, end up going to this carbon and this nitrogen end up going to make N5 form amino FH4, and these guys go to glutamate. Glutamate is how many carbons? 5. So where does it go? It's gluconeogenic. It goes to alpha-ketoglutarate. Okay. And this one we also did, and the important thing about tryptophan degradation is that it goes to nicotinamide. So if you have sufficient amounts of tryptophan in your diet, in the protein in your diet, you can make nicotinamide without having to augment it with niacin on that basis. So, there we go. 3 minutes late, 2 minutes late. Thank you very much. I'll see you. So next week, next Friday, I guess we're going to have a quiz again. Okay. So, by the way, if you have any questions, send me an email.